Hi everyone, my name is Kajal Rani and my partner name is Krunal Pandya. The topic we are going to present is effect of neem oil on E.coli and G.candidum. I'll be covering introduction, method and material, and Krunal will be covering results, conclusion and discussion. It is estimated that during post-harvest handling, approximately 20 to 25% food is spoiled by food decaying bacteria worldwide. So, G.candidum is one of the fruit rot fungus that is responsible for the decaying of fruits such as guava. Apart from that, it is also associated with the spoilage of dairy products such as cheese, etc. E. coli can be commonly found in human gut and the intestines of warm-blooded animals. In case of food, it is associated with the hamburger disease that is caused by the consumption of ground beef patties. Now, coming to the neem oil, it is one of the ancient way of treatment. And in case of food, it possesses antimicrobial properties against E. coli because of the biological active compounds it has, such as nimbolate. So in this experiment, neem oil at different concentrations was used against E. coli and G.candidum. In case of hypothesis behind this experiment, it was associated with the dilutions of neem oil. And according to that, high dilution of neem oil would be more effective against the growth of E. coli and G.candidum. So to check the MIC, that is minimum inhibitory concentration, bacterial cultural suspension for E. coli and G.candidum were prepared separately, and after that E. coli and G.candidum were suspended in 5 ml of TSB and SDB tubes respectively, along with the diluted neem oil diluted at different concentrations. In order to start the experiment, the master plates of E. coli and G.candidum were grown in the lab. In case of E. coli, TSC plate was used, that stands for Triticase soy agar. Similarly, G.candidum was grown on SDA plate, that stands for Sabio dextrose agar, and after that, both the plates were incubated at optimum time and temperature. The effectiveness of neem oil against E. coli and G.candidum was checked at different concentrations. And in case of dilutions of neem oil, dilutions were prepared in two diluents. The first one was sterilized distilled water and the other one was 280. And according to the referred research article, 0.1 to 0.5 concentration of 280 do not affect the growth of microorganism. So we did use the 0.3 concentration of 280. In order to prepare controls, the bacterial cultural suspension was prepared in 5 ml of sterilized distilled water, and in case of positive control, for each microorganism, 3 different controls were prepared. The only difference between the positive controls for both microorganisms was media used. In case of E. coli, TSB was used, and in case of G.candidum, SDB was used. Similarly, negative controls for both microorganisms were prepared in their respective media. And as similar to positive controls for each microorganism, 3 different controls were prepared. And after that, the tubes were incubated at optimum time and temperature. The effectiveness of neem oil against the growth of E. coli and G.candidum was determined using MIC, that is minimum inhibition concentration, and it is the lowest concentration of a chemical or drug which prevents visible growth of a bacteria. And for both microorganisms, the first tube of the broth that showed no growth was considered as MIC. So now to check the susceptibility of a microorganism, a guard diffusion method was used, and for that in TS and STA plates, wells were made. And first of all, 0.1 ml of cultural suspension for both microorganisms was spread on their respective plates. And after that, the resulted MIC tube for both microorganisms was inoculated in their respective plates. And then the plates were incubated at optimum time and temperature.
Hello everyone, my name is Krunal Pandya and I will explain the result and conclusion part of our experiment. So basically result section has 3 different parts. So the first section is a master plate preparation. As a result of master plate, we were able to obtain isolated colonies for E. coli and Geotrichum candidum. The colony appearance of E. coli was entire, smooth, circular in shape and convex. while the colonies of Geotrichum candidum obtained on master plate were white, dry, and had a seed-like appearance, which we can see in the picture. So in the second section of result, we will talk about MIC. In the MIC, we prepare two sets. One set contains neem oil diluted with water and neem oil diluted in twin 80. In the given picture, you can see that neem oil diluted with twin 80 on Geotrichum candidum has shown growth on all of their tubes. So we can say that it is difficult to determine MIC for the given results. But this thing is not true for the E. coli, because as you can see, the of dilution 1:32 and 1:64 did not show any growth. So we can say that the MIC for E. coli is 1:32. All the results for positive and negative control tubes were as per our expectations. Yes, both the slide shows the result for the dilution prepared in twin 80. But what about the water? So for the water, we got growth for the all of the tubes for the both the organism. So we can say that for the prediction of MIC, water cannot be used as a diluent. So the last section of the result part is susceptibility test. In that we pour the 1:32 and 1:64 dilution in the well to and allow it to diffuse, and as a result you can see that we did not get any zone of inhibitions, and we can see growth of the organism in all over the plate. So that was end of our result section. Now we will discuss those results and the possible responsible reasons for the error. So the hypothesis of our experiment was that the highest dilution of neem oil should inhibit the growth. From the results, we can say that the hypothesis is not proven, but that would be unfair to say that the hypothesis is wrong, because some experiment has been done and the results of those experiments shows that there is some effect of neem oil on E. coli. In 2017, there is one experiment which shows the MIC of neem oil for E. coli was 1:32 and their zone of inhibition was 22 millimeters. One more experiment done in Dhaka, Bangladesh, which shows the similar results for the E. coli. In case of Geotrichum candidum, there are very less evidences available. One of the evidences from the experiment which was performed in 2014, which shows that the minimum 30 to 50% concentration of neem oil required to inhibit the growth of the fungi. Less than those concentrations like 10% or 20% would not be enough to inhibit the growth. There can be few reasons for the obtained results. The first one is the presence of the dead organisms in the tube. In the tube of initial dilution, turbidity may because of the dead cells. So in that case, we can say that the concentration was enough to kill the organisms. Second is the density of the diluent used. We use two diluents, twin 80 and distilled water. Twin 80 was too dense, which may not allow the proper distribution of the neem oil, and this can be the same case for the water, but water is very less dense, which affect the distribution of the neem oil. The third and the final reason could be the component of the neem oil. Neem oil which we used here in this experiment was for the retail sale, so during the processing there might be some component added which affect the effectiveness of the oil. After looking at the possible sources of error, now it's time to see what can be done to get proper results. One is to ensure quality and density of neem oil. For that, extraction and filtration process of neem oil should be done in the lab before the beginning of our experiment. The other thing which should be taken care to get the proper results is to set the controls properly. Here we did not set the control of twin 80, so we cannot say that the twin 80 does not have any impact on the growth rate of the organism. So, to conclude, we can say that we were not able to prove the hypothesis due to some reasons which were explained in the discussion, but it is possible to prove the hypothesis by taking care of those aspects.